all of us here are hoping for treatments and cures, and many of us want to participate in getting there however most expeditiously we can. We would like to magically say, here is the treatment or the cure, but it's not an instant thing to get to that goal. So how can we do this better? And that's what I'm going to talk about today, is the information and the studies that happen before a treatment or a drug trial. And that is what biobanks and natural history studies are. So where do we begin? It can be a really long path. Some drugs can take a decade or more to go from an idea at the bench to a treatment and to be able to write a prescription for it on the market. The success rate is not necessarily high. Only about 1 in 10 clinical trials is successful, and it's even lower for things that have central nervous system involvement. And while the FDA promotes rare disease drug development with the Orphan Drug Act, there's so much that has to go on before a drug even comes before the FDA for approval. So one of the things that we want to talk about today are, what can we do to be faster and to be smarter and to be more successful in developing treatments? So first, we need to look at the ingredients to what makes up a successful treatment trial. Some of these things are knowing what the drug target is, knowing what change we're looking for, how long should the study go on, how many people are we going to need, and where are we going to find them, and what age are the subjects going to be, and are we looking at a particular severity of symptoms or duration of symptoms, and are there people who shouldn't be included because it might be risky to them to be in the study, and how do we get that information? The only way we can get that information is from you. So I'm going to start with the math, but I'm not going to get too deep into it. Basically, in order to develop a good clinical trial, you want to be right. You want to have a good chance of being correct. And that means what we call a true positive. You don't want a false positive. That's something called a type 1 error. Or you don't want to miss finding something that was effective and think that it wasn't. That's a type 2 error. So how do we design a study to minimize the errors and optimize the correct responses? One of the ways we look at that is something called a power calculation. And if you like mathematical symbols, it's at the bottom of the page. And another way of looking this is sample size, which is the number of people that you need to enroll in order to minimize a type 1 error, which is thinking you got the right answer, but actually didn't. So what steps will this take? So we're going to need to know, how big a difference are we looking for? In other words, what's the difference between being treated and not? And in order to figure that out, we also need to know what the standard deviation is. And the way I think of standard deviation is everybody who has myotonic dystrophy is very different from each other. There's a lot of variety. Standard deviation is a number that describes that variety. How much difference is there even within one population? And I'll show you some graphs later that kind of illustrate what that looks like and that means. But for now, please humor me with my stick figures. This is literally drawn on a whiteboard to kind of illustrate this, that it could be any outcome measure. It could be cognitive, it could be sleep, it could be fatigue, it could be pain, it could be muscle strength, it could be something, but something that we're measuring, either subjectively, your report of it, or objectively with something that we've measured. And we need to look at this over time. And 0 is the start of a study. Everybody looks the same. And then over time, you start to see differences. So at month 3, those blue bars show that there might be a lot of variation. We can't really tell one group from the other because they're still very similar. But you can see later on, there's more distance between those paths. And you can see clearly there's a difference between these two groups. And we've already decided what difference we're looking for so that we can say we've achieved the goal of testing whether or not this treatment worked or not. So in order to get these numbers, these blue bars and arrows and deltas. We need information from all of you. So how do we start? Where's our beginning point, our starting line? I think the first step is connecting, and we're doing that here today informally, but doing it formally through a registry is a great way of staying connected. Even if you can't be here in person, if you're virtual or you missed this talk and you're watching it online later. Registries are a way for you to connect with a researcher and for the researcher to connect with you. It allows them under, ethically and with IRB approval and all of the committees and boards that are required to do proper research, for you to give us your contact information and for us to get back in touch with you. So you might get contacted for a questionnaire or a survey. And you also might be being contacted to get newsletter updates to say, this is what we're doing. This is what we used your data for. These are the other things that we're doing in our group. You might also be asked some important questions to kind of learn a little bit about you, such as, when was the onset of your symptoms and what symptom was that? What type of symptoms do you have? And to describe that in a general way so we know if you are the right group of people to be in contact with about a study. You may also be asked to give consent to let us review your medical records so we can look back and make sure that we can learn from that as well. And this is called a case series or a retrospective chart review. And there's a lot of data that can be mined from this. And it also often will allow us to share de-identified information, not your name and phone number, but grouped aggregated data with other groups who are interested in developing myotonic dystrophy treatments. So this is really important. Information to other researchers, information with drug developers to know what's feasible. So here are some registries. This is far from all-inclusive. The earliest registry I know of is the Rochester Registry that was started in 2000 by Dr. Moxley and Dr. Thornton. It has had over 30 projects that have come out of this study. So this information does go places. The Myotonic Dystrophy Foundation also has a registry that I think has thousands of people enrolled. And it is also additionally used for advocacy. This information will be used to help raise awareness and funding for myotonic dystrophy research and treatments. It also, I'm really excited about this, has a view data tool. So once you're signed up, you can kind of explore what has been put in by other people and learn about the data that's being accumulated. And it's not just in the United States. For example, the United Kingdom has a myotonic dystrophy patient registry for both type 1 and type 2. And it has produced 32 projects and 31 publications to their website when it was last updated. But none of this rules out you enrolling at your own local institution so your doctor can get back in touch with you. We do not have perfect photographic memories to remember each of you and what your symptoms are when a particular study comes forward and says, hey, do you have potential people who are interested? It helps us gather the information so we can be back in touch with you. And the other wonderful thing is this is non-exclusive. You can sign up for all of these. Well, I'm not sure about the UK if you don't live in the UK, but it is okay to be in more than one registry locally and nationally to stay informed. Okay, what else can we do to gather information? One thing that you can do is be part of a focus group if you hear of one or are aware of one, and to take part in interviews. So here are some examples of things that have come out of focus groups. One is the development of patient-reported outcome measures, and Chad Heatwell has developed a number of these that are excellent measures of how myotonic dystrophy affects people and what its impact is on their life. This has also been done in myotonic type 2 and also for congenital myotonic dystrophy. Another important thing are focus groups and workshops. So some of these have been hosted by the MDF over the years. This is a great way of amplifying your voice. You talk with your family, with your support groups, with each other at these meetings, but whose ear do we need? And that's one of the things Kevin works on, is getting the attention in the ear of other researchers, of NIH, of the FDA, of government representatives, to know the importance of myotonic dystrophy research. So if you can be part of one of these workshops, 2 of which are listed here and are published, it really helps amplify your voice. So you might feel like questionnaires and surveys are very mundane, and they take a lot of time, and they're asking a lot of fiddly questions. But we learn so much from them. And we encourage you to take part in any that you might hear of that you're eligible for. One example of this, and you probably have seen some booklets of this lying around at the meeting, is the report from the Christopher Project, which was funded by the Marigold Foundation. And one of my colleagues, Dr. Hagerman, helped analyze this data. And it tells us not only about the prevalence of particular symptoms in the different groups, but also its impact score. And you probably can't read this slide because the print is rather small. But what I wanted to point out is that, yes, muscle weakness is very important. But look at the relative report of the importance of things like fatigue and daytime sleepiness. And going down the list a little further, things like muscle aches and cramps and pain. And then cognitive things like difficulty concentrating, difficulty thinking, mood problems like anxiety, gastrointestinal problems, and swallowing problems also are very common and very prominent. And so this really emphasizes and highlights how multisystemic these disorders are. So these patient-reported outcomes can be used as an outcome measure, such as what Chad Heatwell has developed in clinical trials as a patient-reported outcome. The NIH wants to hear your voice, your opinion as to what is important to you. And this is one way of measuring that. And this has highlighted the importance of symptoms such as fatigue, sleep, and gastrointestinal symptoms. So what else can we do to get as much data as fast as possible? Well, one thing is to look back. And what I'm trying to illustrate in this is the difference in gait and stride and shoe size and all of these, the variety and richness of experience that happens in people who have myotonic dystrophy. And following people over time can tell us a tremendous amount. So here's an example from Dr. Bouchard's group that looks at hand grip force over the years. And you can see that it declines over time. But another thing I want you to notice about this, and I don't have a pointer for those of you on the webinar, but if you look at the 10 to 20 year range, you notice lots of dots with a lot of vertical scatter. So yes, we have a lot of people, but look how spread out it is. We might look at the later age group between 40 and 50 and say, oh, look, lots less scatter. That standard deviation I talked about earlier is going to be a really narrow number. So we're not going to need a lot of people. But then look at how few people we have in that age range. What does that mean? It means we need to gather more data in that age range, that if we go from having 4 people in the 40 to 50 age range to 50 people in, like we do in the 10 to 20 age range, that standard deviation might change. That's going to change our calculation. That's going to change our sample size. That's going to change our chance of success in that clinical trial. So if you only look at 2 or 3 people and you think that your standard deviation looks great, it might not be the case when you start enrolling more people, more different people, more varied people, and that power might go away, and we might make one of those type 1 or type 2 errors. So here's an example of something that has a nice standard deviation profile. You see how these dots all like to stick to the line? We like to see that. This is an example of looking at back through time at EKG changes of the heart. So an example of a different organ system, also very important, also really important to see what the changes are over time. And they found there's a correlation with your CTG repeat size, which might be something, if you know it, you'll be asked in registries, questionnaires, and enrollment in studies. But we also want to look forward. So looking backward is nice, but it might be a little bit biased. We might not have been asking the right questions. Not all the data may be collected and complete. So how can we ensure that we've got the best quality data possible? It's to look forward. And so these are prospective studies. And so here's an example from Cynthia Gagnon's group, looking at a 9-year follow-up of upper extremity function. And so you can see that over time, you can see trends, but it might be something that might not be obvious within a 3-month or a 6-month time period. So what else can we do to get more information faster? Well, let's use computers. Let's use big data. Let's mine it for all we've got. Let's use artificial intelligence if we can, and machine learning and whatever technique we can. Here's a study that actually didn't need to employ artificial intelligence, but employed a really wonderful network and system that exists in Utah. And this is work done by Dr. Johnson, and it used the Utah Population Database, which merged data sets of birth records and death records and marriage records and driving records and census information and voter registration, hospitalization information, cancer registries, and payer claims, the insurance companies. And you get really rich data with this. It's not all inclusive, but you can learn so much. And he found that there is an important and increased incidence in central and obstructive sleep apnea. Hypothyroidism is something that needs to be watched and tracked. Intellectual disability is an important factor. But look at this, 60 times increased risk of cardiac conduction disorders. So this is the kind of thing, if you can tell that to a clinical trials group or your legislator, you know, that is such a striking finding. And we wouldn't have been able to do that without this type of data. Here's another example. That was some research done by the Marigold Foundation and Sarah Howe that similarly looked at a very large number of people with myotonic dystrophy, 8,541, and 242 congenital myotonic dystrophy patients with matched controls from millions, 200 million health records over a 4-year period. And here you see a similar pattern to what you saw on some of the questionnaires of what people reported from their own experience. They are seeing their doctors for a lot of gastrointestinal problems, cardiac dysrhythmias, sleep problems, and then we get down to disorders of muscle. And then it carries on. There's more other things, breathing difficulties, urinary difficulties, and then some more general symptoms such as malaise and fatigue, which basically is the daytime sleepiness and fatigue coding. It often gets mushed together. So what are some ways that we can prospectively measure high quality, you know, not a lot of missing data, capturing the sorts of things that we've learned from you are important going forward to design these trials? Well, these are the natural history studies. And one of these was just discussed today in the professional track, were some initial findings, and Dr. Johnson can, if he's here, you can ask him more details as well about what was found with the previous DMCRN study and the ongoing NDM1 study, looking at changes in things like walking speed, your lung capacity, cognitive function, and also getting these important biomarker materials like needle muscle biopsies to look at transcriptomes that you might have heard about in these different sessions, and blood samples for other biomarkers. And there is probably someone near you who's a center. You look at this long and lengthening list, both in the United States and in Europe, for potential enrollment. But there's many other teams and many other networks that have led to really important discoveries and the type of information we need for good clinical trials. The Optimistic Network is one, and then which did a study of exercise and cognitive behavioral therapies in an intervention. I want you to remember, not every treatment is a drug. Sometimes it's a therapy or an activity or a behavior. And it found that these were beneficial. And that is a factor that we're going to need to include in our future clinical trials, is asking what type of exercise you do so that we can control, include that as a factor in the study. Another thing that's really important are biobanks and repositories. Sometimes we use these terms interchangeably. They do mean similar things. But basically this means giving material, genetic material, your DNA or your RNA, and this might come from a blood sample. Serum samples are important. We are also interested in collecting spinal fluid because we can learn so much from it. If you've previously had a muscle biopsy, it's possible that sample is still sitting in a freezer somewhere in a pathology department that might share it with the biobank, or you might be able to participate in a needle muscle biopsy. Other surgical tissues might be really important to research. And we can also do cell cultures. Skin cells, which are called fibroblasts, can be grown in a dish, which is what I tried to illustrate there. And then once you have fibroblasts, you can turn them into other cell types like iPSC cells. And then you can turn them into other cell types. So literally you can take a skin cell, turn it backwards into a stem cell like cell line, and then forward again into some other type of cell like a neuron or a muscle or a heart type of cell. There are some multi-disorder national biobanks that, have a little bit of a fee-for-service model, but they are, for research that have collected some samples for myotonic dystrophy type 1 and type 2 of these different cell types, but there aren't very many of them. So for example, they only, when I looked in their catalog, only have 9 samples for myotonic dystrophy type 2, and they only have 37 samples for myotonic dystrophy type 1. So why are these materials so important? You might think, well, why can't you model it in a dish or on a computer or make a mouse? Well, all of those things are also happening, and they're also really important in moving things forward in parallel. But what's the truth that we judge those models against? It's you. It's what we've already learned from you. How well does that model replicate or reproduce your experience as a patient, what we know about the disease? And it might be very specific. It might be the grip myotonia that we observe in humans. Can we observe that in the mouse model? Is there something we can learn about that muscle strength? Is it a heart phenotype? Does it have the brain manifestations that we know are there, or is it just a muscle model? So it depends on what you're trying to study, what type of model you use. And the only way we know a model is good is based on the information that we learn from you. The ultimate gift to us is autopsy tissue, which I know is always a difficult thing to ask about or talk about because it's often at a hard time of life when you're losing or have just lost a family member, a loved one. But there are some things that we can't learn any other way than from autopsy tissue. And to make things easier on your family member, if this is something you're interested in, and it's not for everyone, if it's something you're interested in, contact either our registry or a registry that's doing this. Specify what you want to donate because you get to decide what's done with your body. But most importantly, talk with your family, because after you've passed and you've moved on, it's your family members that are making decisions. So whatever you can do to make that an easier conversation for them is important. And I'm going to ask if there's anyone in the audience a little bit later who has been involved in autopsy donation and they're willing to talk with other people about their experiences to raise their hand. And I just also want to say, we hope that it's a really long time before you give us any autopsy material, and that what we do in our research will make that an even longer amount of time. So, so how does this work? The nuts and bolts are family members contact us and let us know that there's a family member who's passed who decided to donate. We try to organize things if it's logistically possible, because we do have to work with local pathologists and hospitals to arrange things. We still need consent from the family, even if you yourself have said ahead of time, this is what I want. We still need the family's okay. We try to do it in a short period of time, so it's as least intrusive as possible. The protocols that we use allow for open casket burials, and then the body is shipped to and from the autopsy. And then afterwards, the pathologist sends the frozen samples to the biobank. These are stored, and then researchers contact the biobank asking for materials. They have to propose what they want and how much they want and what they're going to do with it. And these are reviewed, and then the samples are sent to the researchers. So for example, at Stanford, with our biobank, with the autopsy samples that have been donated, these are all the different collaborative groups that we've worked with. So a lot has been done. Another example, and I'm going to shout out to Eric Wang's work of looking at transcriptomes of, an example of things we could not have learned any other way, is looking at the changes in transcriptome expression in different muscles in the body. He has similar work where he's looked at the brain as well. One thing that you can do that isn't an autopsy is giving a blood sample or a skin cell sample where we can turn it back into stem cells and then forward into other cells. Well, here's an example of that where patient-derived cells were turned into iPSCs. iPSC stands for induced pluripotent stem cells. And then they were differentiated into other cells, basically neurons. And they form these little spheres, and we call them brains in a dish. And it's a really cool and new and innovative model, and it can be used to screen potential drugs and learn about pathways and mechanisms, which is what was done there. So I'm hoping I didn't go over time, but this is just an overview of the wide world of research of how do we gather information through observation and your involvement so that we can get to those successful treatments and cures. And now I'm going to pass it over to Kevin.
Thank you very much, Dr. Sampson. When I was here yesterday and I was sitting down, I saw that there was a flyer announcing our presentation, and I've never had a chance to tell everybody that I was co-presenting with a physician researcher from Stanford. So I sent a text to my twins who are 13 and my daughter. I said, look, I'm a big deal. And my daughter, Paige, who is wonderful and precocious, said to me, sure you are, bro. Sure you are. And I said, keeping it grounded. Well, it is an absolute honor to be with you today. I think it was Jeremy and Eric Kelly who suggested we put the circles on our badges that indicated how many years we've attended and participated in these events. And I've done 8. And it's just sort of amazing to sort of think of the journey that we've all been on together. And one of the things that's really hit me in the couple of days that I've been here with you is I feel just such a tremendous change in our sort of trajectory with our grassroots advocacy. There's new energy. I mean, listening to some of the presentations yesterday from some of the companies that are here and talking about where we are with drug development, having the privilege of spending last night with folks from the SAC and talking about the research that they're advancing. I just am kind of amazed. My personal journey is I was diagnosed with a childhood form of arthritis. And when I was a kid, the course of therapy was how many Motrin can you tolerate in a day? And thankfully, there's been a revolution in biologics. And I really feel that we are on that same path and that there are going to be amazing new treatments out there in the very near future. And it is just such an amazing time to be here. And just the energy that I'm hearing and seeing from everybody in this room is amazing. And I'm just, thankful to be here. So I'm going to get started with a little bit of a history lesson. So this is a little bit of what we've done as an organization. And there's a real tremendous amount of work here to be proud of that you all have helped drive. And it starts with Kayla's presentation that we heard about this morning, where we set ourselves on a path with passage of the MD Care Act, which helped establish Wellstone centers, which we have been a part of that have helped drive research. You can sort of walk through the different changes. I'm not going to highlight all of this in the time that I'm here, but one other important piece was when we talk about the trajectory around research, the addition of myotonic dystrophy in a rare program at the Department of Defense called the Peer-Reviewed Medical Research Program. And that was an area where we built off of some of the research funding that we've secured through the NIH, through the Department of Defense, which has helped fund new and interesting and amazing research. We've had testimony from before Congress. You can see Tim Halen, who was there in 2019, who testified about myotonic dystrophy and the need for research, and on and on and on. So I'm going to move forward and start talking a little bit about where we are and where we're headed into the future. So we've had also some great congressional champions who have stepped forward, and we continue to work with them, and they are so key. We've built the awareness with them, and we've helped motivate them to help us. And you can see Martha Montag Brown, who's our vice chair here with Adam Schiff. We've also have Senator Dick Durbin from Illinois, Senator Feinstein from California, and my personal favorite, Congresswoman Rosa DeLauro, who was my boss back in the day when I was her chief of staff in Congress. So what we want to talk to you about today is how do we raise our voices beyond where we already are so that we can drive even more federal research dollars to do more of the wonderful work that Dr. Sampson is doing at Stanford and some of the researchers that are here today are doing. So the first place I want to start is just a quick overview of the National Institutes of Health. It was established roughly in 1887. At the time, it was known as the Marine Hospital Service. It is the largest biomedical research funder in the entire world. It is the crown jewel of research. They have been funding 168 Nobel Prize winners. There are 27 institutes that focus on heart, aging, childhood issues. It's amazing. And that's a picture right there of the campus. They have a process around competitive peer review where they receive proposals, research proposals from around the country. They award those based on merit. And the agency is annually funded by Congress. Congress writes the check that supports the research. So here's a snapshot of where we are, myotonic dystrophy, relative to some other conditions that you will be familiar with. And you can see that our research line is flat. Okay, so if we stay silent, that'll stay flat. And one of the things we want to do today is talk to you about how to take our voices to make sure Congress understands that this should be a bigger priority. And I used to tell folks back in my previous job when I was with the Arthritis Foundation that, members of Congress just don't wake up in the morning and say, oh, kids get arthritis. It's because we tell our stories in a way that has an impact on them, and that message stays with them, and we continue that message. So we're going to talk a little bit about how we're going to change this number. So one of the things we've done successfully over the last 5 years is when the NIH was flat, we looked at other sources of funding. So the Department of Defense has a unique program called the Congressionally Directed Medical Research Program. This was started in 1992, mostly by breast cancer organizations who were similarly frustrated with flat funding or not as much funding as they felt was being provided. And they went to the Department of Defense and they created this program, which has since grown over time. So we were able to get myotonic dystrophy added as an eligible area of research in fiscal year 2017. And since we've done that, we've had success every year maintaining our eligibility in the program, which you have to do every year. There are no permanent designations. You have to get Congress to do it every year. And because of that success, we've been able to get an extra $8.1 million in funding to support researchers around the country who are doing this important work. So this is just a quick snapshot that shows you sort of year to year what we've been able to do. Fiscal year 22 is pending. That means they're reviewing applications and we'll get a number there, but you can get a little sense of sort of how that tracks. So just to excite everybody here, I'm going to talk next about the congressional funding process, which I know all of you are dying to hear about. And to get started, this is a slide that hopefully you can read. This slide actually used to sit on my cubicle when I first started out on Capitol Hill as a young congressional staffer in about 1993. And it was my job to answer the mail. So any letter that came into the office, I was the guy in the office who answered it and sent you guys your responses. And this always gave me a bit of a chuckle. This is how a bill becomes a law. And while I was a political science major, that's why I got on the panel and research here. I'm a scientist by nature. You can see this is a swing set on a tree, and it shows you a bill as introduced, which is it's got its one thing. And you can sort of walk through how it's funded by committee, how it's understood by the public, which is a blank slide. And then at the end of the day, what was really needed. So I just wanted to add a little levity, and it makes me laugh that I found this from 25 years ago. So what is the budget process? How does it work? And why should we care about this? Okay. So the president gives the State of the Union address in January of each year. It's actually mandated. It has to be invited by the Speaker of the House. Shortly thereafter, the president submits a budget to Congress. Congress takes that budget and they turn it into what's called a budget resolution, which just sets the broader parameters of spending. And that sort of launches then in the spring, the appropriations process, which is where we are focused. So they will bring the NIH, every federal agency before Congress for hearings to talk about the budget. It's an opportunity which we've used in the past to raise awareness. We've actually had members of Congress ask questions of NIH directors about myotonic dystrophy funding. And that's the process where then they start to craft what the final bill will look like and where the budget priorities get identified. That process kind of moves through the summer. The Senate generally follows the House and puts together their own separate budget. And then into the fall, they combine those efforts so there's one unified budget. Now it's supposed to be passed by September 30th, which is the end of the fiscal year. We have been in a state of dysfunction, to put it mildly. So we have tended to operate off of what's called a continuing resolution, which keeps the government funded at current funding levels until they can finally pass a budget, which unfortunately is sometime after the fiscal year. So what are our goals? What are we trying to accomplish? You know, it's great that we've got a budget process, fine, but what are we trying to do? So we've got a couple of goals here to talk about. One is we're looking towards the fiscal year 2003 budget. So we've been working towards getting a new initiative. As I mentioned, we've been stuck at NIH. The funding's been flat. We've got a new initiative here to try to change that, and that is the Repeat Expansion Diseases Initiative. We're also looking, as I mentioned, to keep in the PR MRP program for the 6th year in a row. And we have another really bold initiative where we're trying to get an actual earmark of $10 million in funding for myotonic dystrophy research in the new budget. So let me talk about this initiative. The READY initiative is, I think, one of the most exciting things that the Myotonic Dystrophy Foundation has done this year in advocacy. And what we've looked at is an opportunity driven by Eric Wang, a researcher at the University of Florida and a member of our National Advocacy Committee, in trying to find a new way to approach Congress about funding and using myotonic dystrophy as sort of the lead in an effort to grow this effort. So this language is from the bill that Congress passed in the House in June. And it's the first time ever we are launching this new initiative in NIH. And this is Congress telling the NIH, this is something that we're going to expect you to do. So now that the budget is about, or we're entering a new fiscal year, we're going to be meeting with the Institute directors at NIH and talking about how they can take this new initiative, how we're going to do different and new funding. So this is going to open up a whole new era of funding for myotonic dystrophy research and repeat expansion diseases. So there's more to come here, but we're going to be seeking to continue to build this through our grassroots efforts that I'll talk about in a minute. Mentioned the CDMRP, PRMRP. These are the Department of Defense funding pieces. I've talked about where we are. Right now, what's important to know here is when the Senate passed their budget, or they didn't pass their budget, apologize. They issued their final statement. It's a little bit quirky, on what they're doing this year on this. They have a list of diseases included. We were not included for the 6th year in a row. So there is a real imperative that I'm going to talk about as far as getting everybody mobilized to ask their members of Congress to fix this omission. I've had an opportunity to, as your representative in Washington, to meet with folks on the Appropriations Committee over the August recess and secure some commitments from members who are going to work to make sure that when they have a House and Senate conference to put together that final budget, that we get added back in. But I've been talking to a few other advocates who have deeper relationships with members of Congress to make sure we continue that drumbeat and that they understand that this is a vital and important priority. So I have every confidence that this is going to be resolved as we head into November in the final budget, but we're going to need all of your help to make that happen. So I want to just ask everybody, sort of my old favorite Uncle Sam, we need everybody here to be a part of this. Is the same way that, Dr. Sampson was talking about making sure that we have as many people here involved in clinical trials. We need as many people here to make sure we can fund and grow that research pot. So I just wanted to see if anybody here who's in our National Advocacy Committee who has been participating in our grassroots, if they could just stand up for a second and just give a wave. I want to make sure that you know that they're here, and we're here to answer questions when this is all finished. So I just want to give you guys a little sense of what you need to do to be an advocate. I know that this is, for me, it's something I do every day. It's something I've wanted to do my whole life. I've worked on Capitol Hill. I know these things. That being said, I spent a decade on Capitol Hill, and the first day that I was doing grassroots advocacy and meeting with, a member of Congress and staff, it was terrifying. I had sat on the other side of that table for 10 years and took thousands of meetings from constituents or interest groups. But the first time I had to go and sell an idea, it was really nerve-wracking. So I absolutely understand, where you're coming from. I'm not asking any of you to become a registered lobbyist. I'm simply asking you to tell the story that's in your heart that you live every day that is so, so important. I mean, you all, are subject matter experts when it comes to myotonic dystrophy. And it's really that awareness that I'm asking you to share. I'm here to help guide you as far as the goals that we have on within Congress, how we help execute that. I'm your boots on the ground. But my job is sort of, it's much more difficult unless we have the folks who can share those stories. These members of Congress will talk to lots of people in a week, in a month. The stories that you all have are ones that will stick with them for an extended period of time. And I just think that's just so important. So don't feel that you have to know everything. You just have to have your story and be willing to share it, and we'll help you do the rest. So that rest part would be right now going to congress.gov. And I know many of you will know who your representatives and senators are, but if you don't, this is where you can get started. So I've done some screenshots here using my parents' home address in Baltimore, Maryland. And so this will help you identify. So that's why you'll see that there are Senator Cardin, Senator Van Hollen, and Congressman Mfume are the folks that pop up when you put in your data. So you put in your street address and your zip code. It may ask for an extra 4 digits. The one thing you have to be aware of, in this case, it was a clean, okay, here's your representative, but where the way congressional districts are currently drawn with computers, it might be that your neighbor who lives across the street lives in a different congressional district than you do. So it may take a little extra work. If you don't know and you get a couple of, call the congressional office. It's always great to just ask if you need that extra help. But this is the first step, is just figuring out who represents me. As one of my favorite political inspirations, Tip O'Neill would say, all politics is local. That's where we're starting. So then moving through, I picked Senator Van Hollen as the next step. So you go through that site and you'll see a click for the link to his website. You go into that, and then to the far right-hand side, you'll see contact, okay? So the two things that we're asking folks to do, one is send an email, make a phone call, whatever is easiest and most comfortable for you, okay? So you can find out through that website how to send an email. So in most cases now, Congress has tried to limit the amount of email they get from folks who are outside of their state or their congressional district. So their way of trying to control that flow is having you go through their website. It used to be that you could say, send it through an email address or what have you, but they just get so inundated with correspondence that they have to try to find a way to make sure that it's just the folks who are there, they're there to represent. So you want to start there. So when you go into Senator Van Hollen's site, you've got two sides that I want to highlight. So one is the email site that I mentioned, but some folks, and I know Emily Jones has been leading the way on this and others have too, where they've been actually doing virtual meetings with their members of Congress. Okay, you can also do in person. We're increasingly seeing members now opening up their offices as we kind of hopefully move through the pandemic. But there's an opportunity there through the site that you can see that allows you to submit a request for a meeting. And truly, it is not as scary as I thought it was that one day, and it won't be for you after you do your first one. In fact, I found it to be the most exhilarating experience because you're able to tell your story, you're able to engage in, the basics of democracy. And it's just a wonderful experience. And it's the way to make sure that your member of Congress doesn't forget that there are people in his state or his or her state or congressional district who are living with myotonic dystrophy, and that they should be thinking about that when it's time to consider funding for the NIH or other things that we care about. So then what do you need to do? You figured out who your members are, and you figured out if you want to do an email or maybe make a call or do a visit. So we've provided on the website some talking points, some information that you can use to create a letter, to create an email. So as I mentioned, all politics is local. So the thing that they care about most is your name and your hometown. So they want to make sure that they're taking care of the folks who, every year are or who they represent. And what's important to know too, you're petitioning them, you're selling them on an idea, you're asking them to do something, right? In about 8 weeks, they're going to be asking you to do something, which is to vote for them. And so, remember, you are in the driver's seat here. They can choose to ignore you, but there's risk to that for them politically. So they are incented to help you, do that. Tell your story. I mean, I've heard just in the last 3 days, so many unbelievable stories from folks that are hard to tell, I know, but they're just so important and they have such an impact. So please, use that. As I said, we've got, in this particular letter, we're focused in on our PRMRP request and our CDMRP request, which we're working to include in that final budget. And then finally, you want to say thank you very much. We forget to do that sometimes in all different sorts of ways of life. Congress cares too to be thanked, so that's important. And then make sure you let them know you're looking for their reply or that you plan to follow up to make sure that you're continuing to hold them accountable. And then, the keys to success, as I mentioned, we're talking about emails, phone calls, letters, visits. All of those things are important, but we want you to be finding the thing that you're most comfortable with. So whatever form that works for you is great. Invite your friends and families to be a part of our network. It's growing. There's so many more people now who are getting involved in our advocacy, and it's truly an amazing thing, and it's making a difference. Next week, we're going to be hosting our International DM Awareness Day webinar, and that's going to be on International DM Awareness Day on September 15th. So it's going to be at 2:00. I will be repeating a lot of the same things I'm doing here. That's on purpose. I know that folks who don't live this every day, it's important that we continue to just make sure we explain the basics and we're available to ask questions. And I think that's another important thing to remember too, which sort of demystifies and makes this a little less scary, because I hear this from folks a lot and they'll say, well, what if they ask a question I don't know? And I said, great. Say you don't know, and you're going to get back to them. So it gives you an automatic way to stay engaged with an office. So you don't have to be an expert. You don't have to try to, come up with an answer on the spot. It's perfectly fine to say you don't know, and I'll get you the answer to that question. So I just want to thank you all for being here, for being an inspiration for the work that I'm doing on your behalf. And I'm looking forward to answering questions. So thank you.
One has a virtual comment or question for us, we will take those questions as well. Otherwise, we'll roam through the room. Just raise your hand and someone will be around with a microphone. Please remember that you must speak into the microphone for your questions so everyone can hear you, including our panelists, and so that our virtual attendees can be part of the conversation too.
I know it's your job, but I want to thank you for all the efforts that you do on behalf of us. My question is, can you repeat those acronyms for what we were to request our congressperson to do?
So the acronyms were, I think that you were referring to are start with the first one. It's the CDMRP, which stands for the Congressionally Directed Medical Research Program.
Okay.
And the second one is PRMRP, and that stands for the Peer Reviewed Medical Research Program. And trust me, it took me about 3 years to be able to say those things. So they're a mouthful. I don't know if there were, if those are the two ones that I could think of, but if there were others that I said, because DC is an acronym minefield.
The other thing I just want to tell everybody, I used to advocate on behalf of National Hemophilia Foundation, and the district right next to me was the Speaker of the House's district at the time. And it was so easy to come in and talk with their staff member, who eventually started calling me and giving me updates on what was going on with her funding. And it scared the hell out of me at the beginning, but it became very friendly.
I've worked with people. I mean, I worked with a young woman who had scleroderma, and she went and developed a relationship with Senator Kit Bond, who's a Republican from Missouri. She went on to get married to a cadet at West Point, and the healthcare staffer with Senator Bond attended her wedding at West Point. It was amazing. So I'm not saying that everybody's going to get invited to weddings, but you'd be surprised, you know, the kinds of relationships you can develop and the interest that's there. I mean, these people, despite, you know, what sort of the cynical nature of things. I mean, when I was a 22-year-old and first came to Washington, D.C., it was to do good. It was to try to help people. And so, I mean, you know, it's still there. And I think if you give these folks a chance, you'll find that you've got an open audience.
The sample letter, the advocacy letter up there, that's on the website now, correct?
Yes, it should be. It should be.
Okay, that's all I need. Thank you.
And do we have any statistics in terms of how many individuals this is impacting in the states? Is that on the website too?
I believe so. I was going to hand it to Dr. Sampson, so I figured she might have the prevalence numbers more in her hand. But yes, there should be on the website, not just in the advocacy section, but I think if you go to the, you know, the about section, you're going to see a lot of those statistics. And I would make a point, those statistics are important, but don't lean too heavily on statistics. You know, I think that there's a lot of compelling statistics. I really do. The stories are what matter most. We do talk about prevalence when we're talking to Congress, but that's not the thing they're going to remember. And that won't necessarily be the thing that motivates them.
I acknowledge that we're in the States, but I'm from Canada, and I'm just wondering if there's something like there's the registry, the Rochester and the MDF registry are obviously in the States. I don't know if Canadians can sign up for that. But if there's anything we can do in terms of contacting government to raise funds for a US foundation, I'm just ignorant. I just don't know whether that's something we can do or not.
So I'll pitch in for the registry information. I think it's fine to register for the Rochester and the MDF registry if you're from Canada. We certainly have done clinical trials in the past that recruit patients from Canada, and some clinical trials also help cover travel costs. So as long as we're not in a COVID-like border shutdown, I think you are very welcome to be involved. It's just, I'm not sure if you can register for the registries in UK and Italy and France and things like that, but sign up for everyone that you're eligible for. And then I'm going to pass it on about the fundraising.
Unfortunately, I don't think that there's much influence that Canadian citizens are going to have as far as Congress is concerned. I mean, we will use examples of, in my work, I've used examples of if you're trying to change coverage and you see that Europe or Canada is doing something more innovative in covering a product that we're not doing with our Medicare program, you can use that to help. But that's generally been it. But as far as just direct constituent interaction, it's hard. But I'd be happy to talk to you about ways that you could be a more effective advocate, with folks in Ottawa, in Canada and what you're doing. I'm not an expert on the Canadian, parliamentary system, but I know enough that I can help you think about how to, create and do an effective advocacy program there.
Hi, I had a question. When you present to Congress, do you do anything with a cost benefit analysis? And the reason I ask, in one of the meetings I was in, they said that they used about $350,000 of state services in a year. And I think if Congress understood that a $10 million investment, one person using state resources of $350,000 is a tremendous thing to do. And just if you have any insight into that.
Yeah, no, members of Congress are keenly interested in anything that that would help save money. The trick is always, investing in the research that's actually going to give you, that information. So I think, again, we're leaning more on anecdotal stories about that. But, if the opportunity presents itself or we become aware of research that's been done, I'm going to hand it to the researcher.
No, but I think that's one of the things that big data mining is helping us find is going through the insurance billing records and finding out what the cost of hospitalization is, how much, if it's a longer hospitalization than typical, and the cost of care, because if we can improve people's health, then we would be decreasing that cost of care.
Hey, Kevin, it's Jeremy back here. That's a great question, because we had John Porter from the NIH, he used to be our chief scientific officer, and we asked him that direct question, which is doing a cost benefit analysis, given how much myotonic dystrophy costs the government, why isn't the NIH putting more money into myotonic dystrophy? And his response was, the NIH does not do a cost benefit analysis. The NIH funds the best science. So that in many ways is one of the big things in terms of getting people into the field, with the fellowships that we do, to feed the pipeline so we have better scientists making more grant applications to the NIH. And that is the way we'll get more money from the NIH. So that whole cost benefit analysis does not figure into any of the NIH's decisions.
But it might to the congressman.
Yeah, no, there's no question. I mean, not to get too deeply into the weeds, but there's a part of Congress called the Congressional Budget Office. And so anytime legislation moves or is potentially going to move through Congress, that organization, the economists and folks there will do an analysis, and they will do a 10-year fiscal analysis to determine what's the impact to the federal government. The challenge is, it is just, what is it going to cost? They won't dynamically score bills, meaning you could go to them. For example, I've worked on a project where you've got a treatment for opioid use disorder. And you try to explain to folks, well, if you are effectively treating that, you're going to keep people out of emergency rooms, out of the judicial system. There's all these savings. And if you move a legislation like that, the Congressional Budget Office, for better or for worse right now, does not care what those other costs are. They only, or reductions are. So it's really hard sometimes to make that argument to Congress. And I know it costs a lot of money to do it. That's why, given the resources and what we have, I think the stories to me are the most important. And if we can reflect that in those stories, it's helpful.
Really enjoying the panel. Could you talk a little bit about the larger context of the federal attitude towards investment in this type of research? Like not necessarily myotonic dystrophy, but the overall investment, whether it's NIH or the DoD stuff, is, you know, where are we now and what's the arc? Is it being valued more? Is it being valued less? Just how does the overall picture look over the next couple of years?
It's a really great question. NIH has grown over the past few years. I think that's likely to continue. We do have, you know, a midterm election coming up in a few weeks. That may change control of one or both houses of Congress. And that may have an impact on funding. I don't quite know, to be honest. I mean, we're in a bit of a volatile political climate. But in general, there's been pretty strong bipartisan support for biomedical research. And I expect that's going to continue, but it's, you know, not exactly easy to, you know, to crystal ball that right now.
Thank you. You know, I know elected officials, they struggle because they aren't in charge, but the thing they can do is obviously be ombudsmen for things and also legislate. One effective tool is when they do the right thing is to recognize them for that. Do we have a program where after they vote properly for legislation that we then follow up?
Yeah, it's something that we've highlighted in our webinars and the training that we've been providing is the importance of that, and then providing templates, you know, for just basic email communications or even talking points that you can use to make sure that you do that. And, you know, there's other even, you know, trying to do a letter to the editor or, you know, just social media to let a member of Congress know. You can use their Twitter handle to say thank you for, you know, supporting myotonic dystrophy research. So yeah, it's really important. And I think, you know, for folks that, again, are interested in becoming part of this movement and our grassroots advocacy, we've started with the building of the National Advocacy Committee. And we've, over the course of the year, been sort of learning as we go along. You know, we've got some basic responsibilities that we've decided were for that committee. But we've also then had other people come now. So, you know, we may start to develop, I don't know, even weekly, monthly calls that we can invite folks to participate in who have questions or, you know, want to check in and understand how to do this, because it really needs to be a full-time, you know, year-round effort. And so as folks get interested and have these ideas, I just want to encourage folks to be thinking about that.
Dr. Sampson, you talked about right here, hi, in the middle. Hi, you talked about a number of different types of research. Where should someone go to get started?
As a participant or as a researcher?
Participant.
So I would start with getting involved in the registries. I would let your local doctor know if they're doing local projects, you can get involved in that. Go to the Myotonic Dystrophy website and look at what research is going on there. And then there's also a really and I should have put this in my talk, there's a website called clinicaltrials.gov, not org, not com, gov. And you can type in myotonic dystrophy, type whichever type you have, and it will tell you, and you can break it down by region. You can look at what's been done in the past, what's currently enrolling, what's not enrolling yet, but will be. And you can look at who's doing it, where are they doing it, what are their inclusion, exclusion criteria? In other words, are you eligible? And so those are some good places to start. And all of those will have a contact number of a research coordinator or someone who's involved in that study. So you can call and inquire, am I eligible? Are you enrolling? How do I get involved? So while I have the microphone, I was going to ask everyone in the audience who's been part of a natural history study, an observational study, or done any type of questionnaire or survey about myotonic dystrophy to raise their hand. That's not bad. It could be a larger number. That's pretty good. We should applaud each other. But now I want anyone who's in the audience who's a researcher who's currently enrolling in a natural history observational study questionnaire or survey, who's looking for participants to raise their hand. Oh, so pointing them out. And the last was the one thing I promised to ask, if there's anyone in the audience who's participated in a donation for autopsy for a loved one, family member, and they're willing to talk about their experience, to raise their hand. Thank you. So afterwards or at tea, for those hand raises, please feel free to circulate and talk, because that's how we get things, how we learn. So.
Hi, thank you for a really interesting panel. So I'm relatively new to the myotonic dystrophy community, but I have worked with an organization in the broader rare disease space called the EveryLife Foundation. And with regard to the question about economic burden, the EveryLife Foundation back in 2019 did a study called the National Economic Burden of Rare Disease Study. And myotonic dystrophy was included in that broader study. So it's a long report, but I would just encourage anybody that's interested in that to check out EveryLife. They have a lot of really great resources. They're not specific to myotonic dystrophy, but may be useful.
Thank you for pointing that out. Actually, we do a lot of collaborative work with the EveryLife Foundation. So there's areas where we might be myotonic dystrophy specific, particularly in the research area. But when there's other sort of broadly, connecting areas around orphan drugs, development, things that a lot of organizations have a stake in, we tend to, sign on to letters that we've launched together. And I know that there was, I'm glad you're reminding us of that study. Also NORD, the National Organization for Rare Diseases, is another group that we collaborate and partner with. So there's lots of folks where we, sometimes we chart a path on our own, and there's many times where we work collaboratively with other groups in those efforts. So thank you for pointing that out.
Thank you very much for a very informative panel. I'm also very new to the situation here and to the myotonic dystrophy family. One thing that would be helpful to me to just hear a little bit more about is the Orphan Bill Act. I've heard that referred to a couple of times in the discussion. It seems to be playing a role. Did I use the word correctly? Orphan Drug Act. Orphan
Drug Act.
Yeah. So just anything you could say about that. Thank you.
No, that's a good point. So several years ago, I think it was back in the 80s, if memory serves me, there was a recognition among the rare disease community that, the biopharmaceutical companies were incented to develop, what they'd call blockbuster drugs when you had a large prevalence of a certain disease or condition. But there weren't sufficient incentives for them to do the research and take the risks, frankly, to develop and bring to market drugs for rare diseases. So Congress enacted some tax incentives and other sort of credits that would encourage companies to do that. Now that's been chipped away at over time because there is, a cost to the federal treasury when you're not bringing in, 100% of the tax revenue that you envisioned. So there have been times where there have been, when they'll pass legislation and they have to have what they call a pay for, meaning they have to, either cut a program or increase taxes in order to balance the budget. And so there's been times where they've taken that out of that tax program. So that's another example of where we partner with EveryLife and NORD and others to be vigilant to make sure that we're trying to guard against any further erosion of that, because it's been a really important game-changing law that's helped lead to more research and ultimately treatments, and we want that to continue. So it is something we pay very close attention to and partner with companies and with other patient groups to make sure we do that. So that's a little bit about the Orphan Drug Act.
Thank you all so much for your participation. Let's give our incredible panelists a hand for this great talk, to Kevin and to Dr. Sampson.